As I mentioned, today's speaker is Oscar Westesson. Oscar's work first caught my eye when his advisor Ian Holmes developed a PHYLO-HMM for recombination detection using structural EM for tree reconstruction. More recently, my mind was blown by his very dense 36-page manuscript describing in mathematical terms what he'll be talking about today. Got it right here. I'm very happy to have him speaking today, and his talk is entitled Accurate Reconstruction of Insertion-Deletion Histories by Statistical Phylogenetics. Take it away.
Okay, thanks, Eric, and thanks everyone for being here. It seems like there's a lot of people, and I don't recognize very many of them, which is really cool. And I think one person is actually from Sweden, which is especially cool. But right, so like Eric said, I'm going to be talking about this method for phylogenetic sequence reconstruction, meaning reconstructing sequences on trees, and that there was a fair bit of mathematics that we sort of developed related to this method, and that's contained in this long document that we actually put on the archive. And so if you're really interested in the math, it's up there. What I'm going to be talking about today is kind of a mix of sort of high-level, somewhat nitty-gritty of how the algorithm works, how it's related to other fundamental algorithms in comp bio, and then finishing with some results and simulated data. You know, how does this work compared to other sort of commonly used alignment programs? So before I get started, I just want to mention the people that are involved with this. This was really came about in the heads of Ian Gerten Lunter is this guy at Oxford who works a lot on indel models, and Ben Payton is at Santa Cruz. So Ian and Gerten were sort of thinking about this approach, and then Ben did something similar but more optimized for reconstructing entire genomes. And then Ian and I, in the last year and a half, sort of fleshed out the math and actually implemented this in a more general sort of alignment program. So what I want to talk about is, aligning sequences and more generally reconstructing sequences on trees. And when I'm talking about a multiple alignment, this just means that we take a bunch of sequences that might be DNA, protein, or even arbitrary sequences that we assume are related by a tree. And we want to assign a sequence, aligned sequence to each node of that tree. And that basically means just placing gaps such that the aligned characters in each column are in some sense considered equivalent or homologous. And by reconstruction, we have the additional task of not just assigning sequences to the leaves, but also assigning aligned sequences to the internal nodes of the tree. And these might, here I'm showing actual sequences, but in some cases it's more useful to leave these as wildcards, sort of marginalizing over the possible actual characters that could be there. And so, most obviously, these two are related by a reconstruction being sort of a more thorough reconstruction of the events, so that a reconstruction contains a multiple alignment sort of within it if you don't look at the sequences at the internal nodes. And the key here is that a lot of analyses in computational biology using a multiple alignment are implicitly treating it as a reconstruction. Like if you're using it to score a phylogeny or anything involving substitutions, measuring substitution rates or trying to detect different parts of an alignment that are evolving at a different rate. A lot of these analyses implicitly reconstruct substitution histories conditional on one of these alignments. And so what I want to talk about initially is that most alignment algorithms, if you're making an alignment, you sort of take it for granted in such an analysis. Most of these alignment algorithms don't actually think of an alignment in this way. And there's sort of a subtle difference in that the way that homology is defined in the way these alignment algorithms work is more closely related to structural homology, that different residues that are homologous are participating in similar structural roles. And it's sort of unclear whether this really matters or not, whether an alignment constructed under structural homology is suitable for evolutionary uses. And what I'm going to argue towards the end with some simulations is that this does matter, and that evolutionary measurements based on these alignments will be skewed for the simple reason that the alignment doesn't respect and know about the evolutionary process. And then I'm going to talk about, is there a way forward? Can we construct alignment algorithms that explicitly know about the tree and the evolutionary process and can be run in reasonable time? So the basic outline of my talk is I'm going to talk a little bit about, what is phylogenetic alignment in fairly high-level terms. How does PROPAL work, this new algorithm and program we've developed? And how does it compare to similar programs on simulated data? So this is sort of a sketch of one way to think about different alignment programs, is that they vary in their sort of attention to the tree and the evolutionary process relating the sequences that they're aligning. And I should mention right at the beginning that the two axes, phylogenetic strictness and computation time, are not necessarily either indicative of the accuracy of these programs. So something like ClusLW is slower than muscle and FSA and ProbCons, but it does much worse. And PRANK is more phylogenetically strict in a certain sense, but in our simulations it does worse than ProbCons that doesn't really know about the tree at all. And so this is really just illustrating these two axes. And the general trend is that the more attention you pay to the tree and the more sort of strict your notion of the evolution of the sequences, the more time it's going to take to compute an alignment. And so something like these alignment samplers, Endeline, Bolify, and Statalign, have an explicit model of insertions and deletions and substitutions happening on each branch of the tree. But since exact inference can't really be done outside MCMC, it could take hours to days, even weeks, to compute an alignment. Or something like FSA, ProbCons, muscle, really super fast, but their notion of the tree is sort of gets fuzzier and fuzzier. And then PRANK is sort of here in the middle, that it knows about the tree, treats insertions and deletions differently, and sort of respects the structure of an insertion, but it doesn't actually use a probabilistic model to infer alignments, and this runs relatively fast. So what we were interested in is doing something that was sort of in between these, that had a real probabilistic indel model, but could be run in reasonable time, basically fast enough to reconstruct entire databases or genomes. So that's why I placed the star over here. This is sort of the area of the spectrum we're trying to land in. And one key thing is that we'd like to be able to sort of tailor the accuracy to resource intensity trade-off. So that's what this sort of error bar is representing. I mean, hopefully, if we want to be really, really accurate, we can spend almost as much time as an MCMC sampler. And if we want to just get a quick alignment, or the alignment is relatively trivial, we want to be able to constrain it so that we can run almost as fast as muscle or FSA or something. So that's sort of our goal. Before I get into how we're going to try to accomplish that, I think it's worth just going through what I mean talking about phylogenetic alignment. And so I mean that the sequences that we're trying to align, that I've put down here at the bottom of the tree, A, B, C, and D, and E, we treat these as leaves on a phylogenetic tree, and each of the branches of this tree we assume has some kind of model relating ancestral sequences to a descendant, like, for example, this node evolving to this node under this model. And this is an example of one such model. It's sort of it's called the transducer. I'll get into a little bit later. But it's basically like a pair HMM. It involves sequence evolving according to maybe insertions and deletions and substitutions. And we can make these models as complex as we want. For most of the stuff here, we're using a pretty simple state machine like this. But basically, by phylogenetic alignment, we mean trying to infer an alignment that respects the evolutionary process relating to sequences. And so these transducers that we use to do this, that we put on each branch of our phylogeny, is basically like a PAIR-HMM that's a lot more common in comp bio. The only real difference is that PAIR-HMM models two sequences being emitted from the state machine at once, and the transducer models one sequence coming in, and evolutionary events sort of happening to it, and then another sequence coming on the output. And so, the state diagram over on this side, it looks a bit different than the PAIR-HMM, but anything you can say about a PAIR-HMM, you can say about a transducer, or if you understand PAIR-HMMs, then the transducer is basically the same. So if you prefer to think about it as a PAIR-HMM, that's fine for now. And this is the model that's used essentially by BaliPhy and R2 programs, or Ian's program Handline and my program PROPEL. And so just like you can do, a PAIR-HMM models two sequences, two aligned sequences being emitted, you can do the reverse and do inference under this model. With PAIR-HMMs, you can do it the same way with transducers, just in a slightly different context, in that since they're modeling one sequence going in and another coming out, it makes more sense to think about an unknown ancestor going into each of these machines and generating the two observed sequences. And so the nice thing about this is that, pairwise alignment seems a bit awkward to do in this context because you sort of have to introduce a third sequence. But the nice thing about it is that when you want to put these transducers on a tree, the conditional normalization allows you to sort of plug them onto the tree without worrying about sort of double counting certain sequences like you would with a PAIR-HMM. And so this business about putting transducers on trees, it's fairly tricky, but it is well defined, and it's work that Ian spent a lot of time doing about 10 years ago. And so if you're interested in the details of this, you should check out his 2003 paper in bioinformatics. But sort of just on an intuitive level, you can think of transducers as sort of spitting out sequences, absorbing them, and spitting out others. And so this leads to the notion of coordination of these transducers that are composed on the tree. So here we have one example where we have a 3-node tree and a sequence being emitted at the root, two matched transducers that absorb and then emit sequences down to the leaves of the tree. And we think of the leaves as sort of absorbing and even deleting these sequences that reach it. And the key is that the parents and children of these branches have to be coordinated. So if one transducer is emitting a sequence, the transducer below it must be accepting the sequence, like in a match or delete state. So this is a sort of a legal configuration of transducers on a tree, whereas this, we have something inserting at the root, and then we have another insertion happening on this lower branch and the sister insertion happening over here. And so homologous insertions are not really, we don't think of these as being phylogenetically reasonable. And also this sequence that's being emitted here is not being accepted anywhere. So this is sort of an illegal configuration of transducers. So this took me a little while to really understand. I feel like seeing this movie really made me, and I hope this is coming across okay on the broadcast, but this is sort of a movie of 8-node tree generating a multiple alignment with these transducers. And so green squares are when a sequence is matched and sort of comes in and then goes out. Triangles are either an insertion or deletion, depending on the way it's facing. And so I guess the point of this movie is that we can compose these transducers and coordinate them on any size tree. It's fairly hairy, but it is well defined, and this is basically the foundation of Ian's program Handle and Handleline that try to do inference under this sort of model, either by exhaustive DP or MCMC. We'll just let the rest of this movie play out because it is kind of cool.
So to be clear, that movie, you're just picking one of these states for each site effectively, and then just doing a depth first traversal to figure out what actually the sites are.
Sort of. There is it's slightly more involved in that, but basically an insertion happens at some point on the tree, and then below that more insertions can happen and deletions as that signal gets propagated. And then when it gets back to these stop signs, if all the nodes below a certain node are stop signs, then that node is able to either insert or go back to a stop sign. So it sort of winds back up the tree until something happens. Eventually you get all the way back to the root and another insertion happens.
The stop sign isn't a delete.
No, sorry. So the stop sign is this special state called the wait state, and it means that the subtree rooted at that node is sort of finished doing all of its evolutionary operations, and that its parent can now either emit a sequence or itself become a wait state. So this is fairly tricky stuff, and it is related to how PROPAL works, but I don't want to spend too much time on it. And I really think that watching this movie and or reading this paper and then going back to this movie is the best way to understand this stuff.
Maybe you can post the link to that movie at the end of the talk.
Okay, sure. Or actually, it's at our website, biowiki.org. I think the page is called PHYLO, PHYLO, FILM. It has a bunch of these evolutionary movies, and this isn't even the coolest one. There's ones that are like sequence evolution happening and then the whole tree spinning in 3 dimensions. It's really, really cool stuff. But so anyway, going back to this, we can compose these transducers on any tree, and this was happening sort of in forward simulation mode. And the problem becomes that doing it in reverse inference mode, the state space that you have to consider gets really, really big. And so if you have a machine like the transducer I showed in the beginning that has n states, B branches on your tree, then you have b to the n composite states that you have to consider. And the reason for this is that in making one of these composite machines, you're essentially creating a state for all the different evolutionary scenarios that could have happened. So all the possible ancestral sequences and insertions and deletions that could have happened, you sort of have to make a state for each of those and then sum over all of them and do sort of standard DP over a graphical model. But that state space gets so big that even aligning 3 sequences is, of reasonable size is almost impossible. So this is just showing, what the state spaces look like for composing transducers on a 4-node tree, 3 nodes in parallel, and this sort of 2-node and ancestor tree that I mentioned earlier. And so the point of this is just that this is possible, and it gives us a really nice framework to do a sort of genuine indel modeling, but it just isn't really practical for real-world problems. And so, considering this and looking at our possibilities for if we want to do phylogenetic alignment, what are the ways we can do it? And like I mentioned, n-dimensional dynamic programming is basically what constructing that state space and doing inference under it amounts to. You are guaranteed to find the best global ML alignment, but it's basically impossible for time and memory constraints. There have been several approaches doing MCMC using that sort of framework, building an ancestral alignment and then sampling, fixing most parts of the tree, and then sampling, local realignments of a 4-node tree or a 3-node tree. So this is basically taking a random walk through alignment space or reconstruction space. And these do work. And if you run it for an infinite amount of time, it will recover the maximum likelihood solution, and even in reasonable finite time, like I said, hours to maybe a day or two, or a really big alignment, a week, they do reasonably well, but they are very slow. And so if you want to reconstruct an entire database or thousands and thousands of simulated alignments, you start having to actually consider computation cost. If you either need a cluster or rent cloud nodes, and so it gets into the space of having to consider the economics of doing all this computation. So what I'm going to talk about today is this method that we have that's sort of trying to be one step more tractable. It uses this technique of progressive alignment, basically just proceeding up the tree in post order, and these ways of representing ensembles of alignments called sequence profiles. And the global ML alignment is not guaranteed, but it hopefully in quotes, meaning it does a stochastic approximation, and so it's not guaranteed, but if the solution is reasonable, it will find the global ML. And most importantly, it's almost as fast as a typical alignment program, and it's flexible, it's tunable. You can say how many of the possible reconstruction solutions you want to consider. You can sort of constrain that in these nice ways. So this progressive alignment of sequence profiles is sort of the main meat of how PROPAL works. So I'm going to try to go through that as clearly as I can, but like I said, or like Eric mentioned, it took us 35 pages of pretty dense LaTeX to describe exactly how it works. So, but if you do have, if you have questions, please do speak up with the chat box or ping Eric. So I think the best way to describe this is its relation to Felsenstein's pruning algorithm, which is a fundamental algorithm in statistical phylogenetics. And this is basically what allows us to compute the likelihood of a set of sequences conditional on a tree. So if we want to compare different models or use a phylogrammer that has different substitution models for different parts of the alignment, Felsenstein's algorithm is basically the mathematical machinery that allows us to do that. So in a sense, this sort of underpins all of likelihood phylogenetics. And it's based on this fundamental probabilistic idea that if you have something you want to know, but you can only compute it in terms of something else, then you have to sum over all the possibilities that you don't know. And so in phylogenetics, typically we know the sequences at the leaves, but we don't know the sequences at the internal nodes. And so if we want to compute the likelihood of the sequences that we observe, we have to sum over all the possible internal sequences, which seems like this gigantic exponential sum, but with Felsenstein's algorithm we can do it in linear time using this sort of clever recursion. The basic idea is that you summarize each internal node with this quantity over here that's I've denoted Fn of x, which just means you can think of it as the likelihood of the subtree below n, given that node n has taken or has value x. And that's recursion just goes that you sum over the nodes descended from n, their likelihood conditional on n having this value. And so there's two things to note here. Is that this conditional probability is basically where the evolutionary model comes in. So you can say you condition on something happening at this internal node. You say, okay, what's the probability that this evolved into its child? And that's where your evolutionary model sort of enters in this recursion. And there is some, there are the boundary cases of computing this quantity for leaves in the root, but they're fairly intuitive. I'm just going to show this case for now. And so the key is that conditional pairwise model that has been used in this, for its typical use cases is a substitution model, something like Jukes-Kenner or HKY or Kimura or whatever, that describes one character mutating into another. And so what that amounts to is that if we want to compute the likelihood of an entire alignment, we have to compute and store this quantity for each internal node and for each column of the alignment and for each character. And then if we make the assumption that each column evolves independently, we can just compute this for each column and then multiply them all together, and that's where we get our likelihood. And so that's the case when we assume that X is a character, like a single character in a given column. And so we do have to compute, or we do have to store this for all X, and when all the X is just the letters in our alphabet, this is not such a big deal. But what we're going to try to do, or what PRUPAL does, is actually try to do this for sequences. So assume that X is a sequence, and this conditional probability is not just a model that describes one character evolving into another, but actually one sequence evolving into another. And so if you remember, that's basically what a transducer gives us. It's a model for, given that one sequence is coming in, what's the distributions of the distribution of sequences coming out. And so just on an intuitive level, it seems like this is not possible because, if we want to store this quantity for all X, where X is sequences, then it seems like we'd have to store infinitely many possibilities. There are infinitely many sequences for a given alphabet, if the sequence can have any length. And so if you're having kind of a weird disconnect, that's certainly valid. So what I'm going to try to do now is go through how we sort of first approximate this, or first, why that space is not exactly infinite, but it's still very big, and then how do we sort of approximate that very big space. And that's basically the guts of our PRUPAL program. So what we're going to use here is that exact inference for a pair of sequences with a pair HMM or a transducer is tractable. We can align two sequences and actually compute the full posterior of the alignments or the reconstructions relating them. But that doing that for a whole tree, whereas for substitutions that can be done in ln time for n sequences of length L, it's very big for if you assume that each of those units is sort of a sequence. And the key sort of mathematical or algorithmic tool that we're going to use to do this, that sort of allows this whole thing to happen, is this set of nested approximations, where we sort of compute the full posterior of aligning these two sequences and approximate this internal node here, represented by this cloud. And we do the same thing over here, make a cloud for this node, and then joining the clouds is actually fairly similar to joining sequences. So this is kind of a complicated looking graph, but if you are familiar with basic pairwise alignment, like the Needleman-Wunsch algorithm, this is actually has the same sort of structure. So there are different boxes here with little dots representing different sort of homology statements. And a typical Needleman-Wunsch table is just has either aligning two characters or a gap in character. The only difference here is that we differentiate between an insertion, where there is no ancestor and a character is aligned to nothing, and when there is an ancestor, we are aligning, again, a character to nothing, but the ancestor was there. And so this sort of matrix of states is equivalent to the Needle language algorithm augmented with sort of insertion and deletion information. So instead of just a pairwise alignment, it's sort of a pairwise reconstruction. And the way to think about it is that any path starting from the start state and ending in the end state is identifiable with an evolutionary history relating the two sequences. So for example, if we start here and we follow this arrow to this green end ancestor state, that's saying that we're aligning the G and the G, and here aligning the I and the L, and then here we explain this extra V by it being an insertion. So there is no ancestor, and there's no corresponding sequence or corresponding character in the sequence. And so that you can think of that as the alignment over here, where the homology statements are here, and then the ancestral character that we leave marginalized, this black character. We don't know what it is, but there is a character there. It's represented by the star. And then when there is no ancestral character, when there's been an insertion, then we have a gap in the ancestor. And so that is sort of the way you would normally do pairwise alignment. And the core problem here is that while you can do this for different leaves or different sort of subtrees of our main tree, the optimal alignment of two sister sequences may not be known until you have all the information or the information from the entire tree. So it makes it impossible to guess what the optimal alignment of GL and GIV is if you don't know the sequences in the rest of the tree. And so we have this conundrum of, we don't want to consider all possible solutions and store them as we go up the tree. But if we only store one, it's unlikely that it will be the correct one once we know all the rest of the information. So that's what motivates using these sequence profiles. So instead of storing all the possible solutions, we try to store a high probability subset of the state space. And storing this result is something we call the sequence profile. And so in this case, it might not have been very clear whether this red V character was the result of an insertion or deletion. I mean, how can you know? We just have GL and GIV if the V was the result of the ancestral character being deleted or an insertion happening on this branch. And so we'd like to store, this possibility that this resulted from a deletion. And maybe these have equal probability under the model. And so the way we do this is that this matrix of states, is a straightforward graphical model. We can sort of compute analogous to the forward algorithm in a regular HMM, sort of probabilistically summarize the whole state space and then trace backwards through it proportional to the posterior probability of a given path. So we can sort of select parts of this state space that are sort of reasonable or meaningful. And so here's our third sample, is that we think that this I character is not homologous to L and that it arose by a deletion, and that the L character similarly arose by a deletion, and then we sample through one of these. So you can see as we add more solutions or more paths that we've traced through, we sort of keep them all stored in this profile. And what these profiles do is that they allow us to encode a sort of a distribution over alignments, or an ensemble of alignments, that allows us to, or this distribution sort of can tell us a lot about which parts of the alignment are uncertain and which aren't. And it doesn't make much sense to store all of these solutions in this part of the alignment if it's very certain. Maybe all of these characters are perfectly identical, and there's no question whether or not they should be aligned, whereas here, maybe more insertions and deletions have happened and we're not so sure of the alignment. So this gives us a nice summary of the alignment space, but it also allows us to maintain tractability for proceeding up the tree. So instead of storing all of the possible solutions, we can store this alignment profile that we can then align with other subtrees and continue up the tree. All right, so like I said, tractability, robustness, it allows us to store multiple solutions. And then finally, somewhat more in a mathematical sense, it sort of gives us this nice self-consistency in that when we're aligning sequences with profiles, we can think of a sequence as being sort of a very simple profile. So the algorithm to align any two objects is pretty much the same if you think of them in this way. And so the way we use these for alignment is that we create a profile for each of the internal nodes, and then we move up the tree, starting from the leaves, going up to the root, and join these profiles iteratively. Like I said, treating leaf sequences as just trivial sort of linear profiles with no branching. And so there's a few little tricky points about this. I'm not going to spend too much time, but basically if we have an insertion like this, treating insertions strictly in a phylogenetic sense means that this character is not allowed to align to anything else in the tree if we believe that it's an insertion. I mean, that's sort of what an insertion means. And so we want to restrict this when we're aligning this profile to the next sequence. And so basically sort of add new transitions and delete other ones such that this likelihood of going from A directly to C, where B is an insertion state, is sort of the product of these two. When the profiles get really big and there's a lot of these transitions, it gets slightly hairy, but since the graph is guaranteed to be acyclic, it's not a trivial problem, but it's also not a really big deal. But like I said, I'm not going to spend too much time going over how this works. But suffice it to say, it's possible to marginalize over these insertion states. But so by treating these insertions strictly, it means that if we make a mistake low in the tree and we say, okay, I'm sure that this red character is an insertion, I'm not going to allow it to align to anything else in the tree. If we make that mistake low in the tree, and then later on we would have liked to align that to something else in the tree, we're sort of stuck because on the one hand, we want to treat insertion strictly. On the other hand, it might lead to a really unreasonable alignment. And so that's just the nice thing about these sequence profiles, is that we can store both of these possibilities. So when we get up higher and we want to align something to the red, if we have the possibility that it was a deletion on the other branch, we are sort of allowed to align something to this. And it carries the weight of an insertion on this branch, or a deletion on this branch instead of the weight of an insertion on this branch. So intuitively we can delay the decision of calling something an insertion or a deletion until we're higher up in the tree. Right, so I already talked about this. So as we're progressing up the tree, we want to join these profiles with other sequences or other profiles. And this happens in pretty much the same way as you would align two sequences. It is contained in our giant mathematical document if you're really curious about how the sort of standard forward algorithm is adapted to the situation. It's in there, but I'm just going to show a quick example of how this works. You can think of it as sort of choosing two of these states and either matching them or having one of them deleted or inserted. And so here we have a green character here that we match to the other green subtree, similarly here. And then luckily enough, we did choose or store this deletion possibility. And now that we see a red character, we sort of do want to match it to here. So we're able to do that because we stored this possibility. And so this sequence of 3 states corresponds to a multiple alignment of all of these sequences, in that sequences or the characters at the leaves of each of these states form the columns of our alignment. And then these black characters, whether or not they're there, sort of form the basis of our ancestral alignment. And so if there's a black character at the root of one of these subtrees, there is a character in that position in the ancestral alignment. So I feel like that was fairly fast description of how this works. And I can't see anybody, and so I'm sort of wondering how lost people are. So if anybody is like totally doesn't know what's going on at this point, there is time for questions at the end, or you can sort of pipe up now if there's something you'd want to have clarified.
I don't see any questions so far. So
okay. So I'm going to continue then and talk about, this is kind of a cool mathematical idea, but multiple alignment has been a central problem for much longer than I've been alive, and so it's it has reached a certain state where alignment programs are really good and they're really fast, and so it's a non-trivial question of whether or not having this sort of strict notion of phylogeny and trying to make an alignment under an indel model will really add anything to the alignment programs that are already out there. So what I'm going to talk about now is results on simulated data, where we basically simulated evolutionary histories on a tree. We wanted to see how well do different alignment programs infer the evolutionary history. And so the way we did this, we actually tried a few different things. And this was, we started simulating with a more basic model, and eventually we started using this external simulation program called INDEL that basically has a simulation model that is several steps richer than what ProPal is using for inference. In particular, it's indel length distribution, the indel composition. It sort of knows about local duplications, and then also rate variation among sites. It could conceivably be built into ProPal, but it isn't at the moment. So we wanted to make this simulation as realistic as possible. And some would say that you should benchmark a program on real data, but the core thing, or the core structure that we're trying to get at here, the ancestral alignment, can't really be, there's not really a good way to have real data that represents that structure. And so, yeah, that's why we went for this simulation approach as opposed to benchmarking on protein structural data, where you sort of know the alignment based on known crystal structures of the proteins, because those they're alignments, but they're not evolutionary or ancestral alignments, and so they're not really assessing exactly what ProPal is trying to do. And so we simulated data on this tree, on the Drosophila tree. We varied the substitution and indel rate over 6 different rates, 6 different indel rates, 3 different substitution rates. And we actually took these indel rates from what we estimated the range of indel rates to be in human genes. So this is about 7,000 alignments that we reconstructed, or sequences that we reconstructed from the optic database that stands for orthologous and paralogous transcripts in clades. This is a database from Chris Ponting's group. It's basically most genes from in the human, together with 6 or so related species. I think it's chicken, mouse, dog, possum, maybe. And so we took all the alignments that were in their database that had exactly one entry for each of those species, and so that we could sort of use the same species tree for all of them. And so one of the caveats with this approach is that you do have to know the tree ahead of time when you're aligning. So looking at data like this was sort of the safest bet. And so what we found is that indel rates are basically gamma-ish distributed with 90% of rates less than 0.1 insertions or deletions per synonymous substitution. And right, so we normalized each rate by the synonymous substitution rate that we computed for each alignment. We found that the rates have this sort of distribution, and so it makes sense to have our simulation sort of cover that range. And a lot of other papers that have done evolutionary simulations have sort of stayed within this range up here, that represents a much smaller evolutionary distance. And in terms of reconstructing insertions and deletions, if you only have very few insertions and deletions, the problem gets substantially easier, especially if they're not overlapping. And so the difficult part of this spectrum is really out here. But yeah, so we endeavored to make our simulation as realistic as possible, both in the program we used to simulate and then also the rates that we fed this program to use. And we found some interesting things. First, it seems like nothing really gets as close to the truth as using the true simulated history, which is sort of obvious that if you have the true ancestral reconstruction, of course, you're going to be able to reconstruct insertion rates and deletion rates very accurately. But on the other hand, protein alignment in general, most aligners are very accurate, especially the top aligners like muscle and FSA and ProbCons. They do really well in these other scores like the AMA or sum of pairs score. But it seems like in phylogenetic alignment, reconstructing evolutionary histories, most programs are not doing that well. And there is this bias towards, so there is this asymmetry in insertion rates versus deletion rates. And I don't have much time to go into it now, but there is a fairly intuitive reason why an alignment program would infer an excess of deletions. It has to do with this stuff I was talking about earlier with, if you call it an insertion low down in the tree, it's fairly likely that you'll be wrong when you get higher up. And so most programs will prefer to call sort of everything a deletion, and that's why we see this in these programs, muscle, FSA, and ProbCons. The histogram is sort of tending to be on the right side of the inferred of 1, where the inferred to true rate is plotted here. And then something we're not quite, haven't quite figured out why this is happening, but FSA, arguably the most accurate alignment program, as measured by these sort of per residue metrics, doesn't do very well on our benchmark. And I think it's the way that FSA treats gaps, in that they're not treated as phylogenetic events, but rather as this sort of abstract gap concept. And then its final trace through the state graph, it just tries to minimize the number of gaps. And I'll show a figure later that is a good example of when more gaps could mean, or fewer gaps means more insertions and deletions, and vice versa. And so gaps are not necessarily the same thing as insertions and deletions. And it's sort of unshaky or shaky ground to make that assumption. And then finally, this program Prank, that was sort of the first program to really make known that using the phylogeny in alignment can have substantial effects on, or using the phylogeny in constructing your alignment can have substantial effects on the type and the quality and the alignment that you get. And in their 2008 science paper, they claim that they had virtually no errors in measuring these parameters, which is true under the parameter range that they simulated, which, like I said, was in that sort of much smaller range, lower indel rate, lower substitution rate than we did here. And so I don't think this discounts the results. It's just saying that as the insertion and deletion rate gets higher, the alignment problem gets harder, and they're not able to really handle those more difficult situations. And I can't get into it much now, but basically PRANK is almost exactly like PROPAL, except that it only stores one solution as it progresses up the tree, rather than storing these profiles of alignments. Yeah. And then our program, is doing reasonably well. It still does have some, the histogram is sort of heavier on the left side, meaning that insertions and deletions are underestimated. And I'm going to sort of present some ideas about why we think that's happening. And we think most of this effect is when insertions and deletions are near each other, either overlapping or very close. And it makes it really hard to infer the true alignment. And so here I have three cases where nearby indels can sort of skew your perception of what the true alignment should be. If they're close together in sequence and close together on the tree, you get this sort of canceling where insertions are underestimated because you have two insertions nearby and you infer 0 of them. And in the second situation, we have an insertion followed by a deletion. And here both rates are underestimated because there was one insertion and one deletion, and you infer none of them. And in this third case, that there's two insertions, but they're far away on the tree. And so if you are trying to minimize the number of gaps, you would pick the right alignment, because there's only 6 gaps here, and there's 16 gaps over here. But if you're trying to minimize the number of insertion and deletion events, which PAL's model assumes that insertions and deletions are fairly unlikely, you would actually pick the left alignment. And so I think this bottom case and generalizations of it are part of the reason why PAL is performing better in these tests of how estimating these evolutionary parameters. Yeah, I think that's all I'll say about that, as I'm running out of time. So I hope I've convinced you that alignment algorithms don't necessarily share the view that an alignment is a summary of the evolutionary history. And I didn't go much into what structural homology is, but the core issue is that alignment programs are typically optimized for these per residue metrics that are really useful if you're trying to predict secondary structure of a protein. You have one and you have an unknown structure, and you align them and you try to infer the structure of the unknown one based on the alignment. You want to have the most number, or the most residues correctly aligned, because that means that your structure prediction will be the best. But I guess the sort of funny thing is that doesn't necessarily correlate to evolutionary accuracy, and that an alignment constructed under that sort of definition of homology doesn't necessarily make a good evolutionary alignment. And that this program and sort of method that we've come up with is one way forward. It's not meant to really replace other alignment programs, but it is kind of sitting in a certain part of the spectrum in that plot I showed in the beginning, that it's uses a explicit model reasonably fast, but maybe not quite as thorough as MCMC, but still doing reasonably well. And not aside from just being an alignment program, it is kind of a nice mathematical framework, and it does have some interesting possibilities that we could sort of extend it to, basically using these ensembles of alignments or profiles for different applications. Like, could we, instead of using an alignment or fixing an alignment and estimating substitution rates or DNDS rates, could we use the whole profile and sort of try to account for the uncertainty of alignment therein? We've been looking at using this sort of framework to estimate tree distributions like pPlacer does, except that the likelihood of a placement would know about the insertions and deletions on the tree and it would know about that the alignment is uncertain. And then finally, in this whole situation, we sort of assume that the tree that we're using for alignment is known, and that's usually not the case. So, it would be possible, it would be fairly trivial to include or to integrate PROPEL with a tree sampler like Mr. Bayes. It's just really hard getting the tractability and the speed sort of up to what it needs to be. But I think with enough cleverness, it could be done. So I think I'll stop there and just acknowledge my advisor Ian and our two collaborators, Hurton and Ben, and let you know that our code is available at this URL.